Okay, now we can start talking about some actions that are taking into account the entire muscle cell at once, now that we know how individual contractions are happening at the molecular level, essentially, and we know how we're turning action potentials into those contractions. We can start talking a little bit more about complete muscle cell actions. And the first thing that I really need to tell you is that, well, twitches are slow. Now, a twitch is just a muscle cell contraction that is resulting from a single muscle action potential. And these twitches are all or nothing. It's you have a twitch or you don't. And really the reason for this is because not so much that there's anything all or nothing inherently about the contraction, but these action potentials are all or nothing. And so the result of these action potentials are really hard. From a single action potential, you really can't get anything different than what you get. So we're going to talk about how this changes a little bit as we start adding up action potentials, but from a single action potential, what you get is an all or nothing twitch. And when we talk about twitches being slow, I mean, in the grand scheme of things, they really aren't slow. We're still talking about something that takes a fraction of a second. However, that being said, they are slow in comparison to the action potentials that are initiating. So muscle action potentials, these are going to last about 2 milliseconds. Very, very quick. Well, one of the reasons that these twitches are slow in comparison is that this excitation-contraction coupling, the coupling between these action potentials and the actual contractions that they result in, is going to take some time. Oftentimes the contraction hasn't even started by the time that the action potential is over. And so we have this lag time in between. And that lag time between the stimulation and the contraction of this muscle cell is what's known as the latent period. And for many muscle cells, this is going to be somewhere in the neighborhood of 2 to 3 milliseconds. So we actually have this delay between the onset of stimulation and the beginning of this contraction. Well, that's not the only thing that makes twitches slow. It's actually the twitch itself that's pretty slow as well. So once a twitch is started, we have several different phases of the twitch. And the first one is this thing called contraction time. Contraction time is the time from the beginning of the twitch to the peak intensity, or the peak kind of contraction strength of the twitch. And that's going to take somewhere in the neighborhood of 50 milliseconds. Accordingly, at the end of the contraction, we also have what's known as relaxation time, which is just the other side of this. It's the time from the peak tension, or the peak contraction strength, to the end of the twitch. This also is going to take somewhere in the neighborhood of 50 milliseconds. And so, therefore, we have an entire twitch that's lasting somewhere on the order of 100 milliseconds. This is like 50 times as long as the action potential took that stimulated this twitch in the first place. So this is going to be an important concept, or an important principle that's going to lead into other things that we're talking about later on. That these twitches, they're taking longer, much longer, than the action potentials that created them. And so it turns out that this becomes an important property when we start talking about, well, what happens when you start to get lots of action potentials. What does that do? So the other thing that we need to complain about twitches for is, even though they take a really long time and they're slow, well, in other ways, they don't really last long enough. So the twitch of a single muscle cell is really too weak to be useful. It doesn't produce enough force to really do anything that is really important for the action of an organism. Any multicellular organism, anyways, it's just really not enough force. And a single twitch doesn't really last long enough to be useful either. So, in the end, the single twitch doesn't really get you much of anything. Well, how do you solve that problem? It turns out that to get around this, we have to add multiple twitches together to get any sort of useful force out of these muscle cells. And there's a couple different ways that we can be adding these twitches together. And so the first way is that we're going to add together twitches of many different fibers, many different muscle cells. And so as we add together the twitches of multiple cells, we get a larger contraction that is much greater force than any one individual muscle cell could produce on their own. Well, that's fairly intuitive. Also, similar to what we were talking about earlier in the nervous system, about the summation, the temporal summation of gradient potentials, we can also get temporal summation of twitches in the same fiber. So if it's stimulated rapidly enough, we can get temporal summation of these twitches in the same place, or in that same muscle cell. So we're going to talk a little bit more about what's going on specifically in each of these individual types of summation. So first up, let's talk about adding these fibers together. And so fibers, or muscle cells, are combined together to make muscles. We talked about this a couple lectures ago, a couple videos ago, about how we add together these muscle cells into muscle fascicles, and then these muscle fascicles are added together to make individual muscles. And so that is how we're adding together these individual fibers to create one larger structure that's going to be able to make contractions that are more worthwhile. And just a couple principles here that we need to understand before we start understanding how these are added together is that one fiber is going to be innervated by only one motor neuron. There's only one motor neuron at any given time as having any influence over a single muscle fiber. And so this means that we don't have any confusing or multiple inputs that need to be summed together in this one muscle cell. It's only listening to one phone line. It's only listening to one single efferent neuron telling it what to do. On the other side, however, is that a single motor neuron is going to innervate more than one muscle fiber. So any efferent motor neuron here is actually going to be synapsing onto quite a few different motor neurons and being able to cause all of them to contract together when it sends along a signal. And so what this means is that these fibers are going to be organized into these things that we call motor units. And a motor unit is simply one motor neuron and all of its associated muscle fibers. And because all of these muscle fibers are linked together by this single motor neuron, and whenever that motor neuron depolarizes, it's going to send the same signal to each of these muscle fibers. It means that muscle fibers in one motor unit are going to contract in synchrony. In effect, they are going to contract as if they are essentially one fiber at the same time for the same duration. And so motor units are essentially these groups of muscle cells that are going to act together in the same muscle in synchrony and essentially have the same contractions at the same time. Okay, so let's kind of look visually what this means. So I've kind of given you an illustration here. Each of these cylinders over here on the left-hand side, each one of these is a muscle cell or a muscle fiber. Each one of these is innervated by a different motor neuron, and I've color coordinated them as such. So for instance, we have this blue motor neuron, and again, like most efferent neurons, it's going to be originating in the gray matter of your spinal cord. It's going to exit the spinal cord via the ventral root of some spinal nerve, and it's going to travel out to some muscle, where it's going to have these multiple axon terminals that are going to synapse onto multiple different muscle fibers. And so, essentially, this blue one is creating this motor unit with these 3 muscle cells and this one motor nerve, or motor neuron, excuse me. We also have a red motor unit here with this one red motor neuron synapsing onto these 3 red cells, these 3 red muscle fibers, We have this green one that's synapsed onto these 3 green muscle fibers. And so each of these motor units can be acting somewhat independently depending on how their motor neurons are firing. But the motor units can act independently from each other. However, muscle fibers in the same motor unit are going to act in synchrony. So all of these blue muscle fibers are going to be contracting at the same time, all the time. And so that's what we're talking about when we talk about a motor unit. And that's the fundamental unit of how we are adding together or summing together contractions of multiple muscle fibers to give us a more useful and sustained muscle contraction. Just to kind of give you another visual of this is a picture I showed you in a previous video here, but again, this is just showing you another example of a motor unit. See this cell right here and this cell right here both being synapsed onto by this same nerve. And so these 2 muscle cells are going to be in the same motor unit. This looks like this is 2 different axons here. Now remember that axons are generally going to be bundled together by similar origin and similar innervation together in nerves. So I think what we have here is one axon here and another axon here. So I think this is a different motor unit. And yeah. One thing to kind of notice here is that these motor units, the muscle fibers for a motor unit, are going to be spread out throughout the muscle somewhat. The reason for that is so as we recruit more and more, or bring online essentially more and more motor units, we don't have muscles that are contracting all on one side or all on the other side or something like that. So the muscle fibers for a given motor unit are going to be somewhat spread out throughout the muscle. They're not going to be all together in one single place. So let's talk a little bit about how these motor units are going to be used in these muscle cells. So first off, each muscle is going to be composed of many, many, many motor units, especially large ones. If you think about any of the large major skeletal muscles in your body, for instance, your biceps, your quads, your calves, anything like that, they're going to have thousands of motor units in them. Now, when you need a weak contraction, or just a light contraction, only a few of these motor units are going to be activated. Only a few of them are going to be used. However, as we need stronger and stronger contractions, we are going to be using increasingly more and more motor units to get a stronger and stronger contraction. And this is a phenomenon known as motor unit recruitment. So we say that we are recruiting additional motor units to get these stronger contractions. And each individual motor unit that we're recruiting, because there's so many of them, and because there's each individual muscle fiber is only giving you very small amount of contractive force, even when we add several of them together, this means that each motor unit is only adding a small amount of added tension, or added contraction strength. But this is a good thing. The reason this is a good thing is that this enables some very fine control over the tension of the muscle. So if you think about this just for a second, you can go out and you can grab something like a styrofoam cup, and the muscles in your arm that control your fingers have by far much more force available to them than is needed to crush a styrofoam cup. It's relatively very easy, but it's pretty simple for a human to be able to pick up some sort of cup, like a styrofoam cup, and not crush it every time. And the reason is because of this fine control that we have over the tension of our muscles, because we only recruit the amount of motor units that are needed to get the amount of tension that we're desiring. Now, there's another thing going on here, is that we also have what's called asynchronous recruitment. Now, most contractions that you have going on at any time in a muscle it's not going to require all of the motor units. It's just going to require some subset. It's very rare that you have some sort of contraction that's going to require all the contraction force that's absolutely available to you in that muscle. Now, when this happens and during sustained contractions, the central nervous system can alternate what motor units are used. So let's imagine us holding this styrofoam cup again. And let's say you're talking to someone, and you're holding this styrofoam cup because you want to drink from it once you're done talking, but you're like me, and you're one of those people that talks for a long time. I tend to do this. I'll just keep talking and talking. Well, that means you need to hold on to this styrofoam cup for an extended period of time. And it's not requiring all of the motor units that are in those muscles to maintain this contraction. So what your central nervous system will do, let's imagine that only takes 10% of the motor units of those muscles to maintain this contraction. What's going to happen is over time, it's not going to be the same 10% of motor units from one moment to the next. Some of them are going to be turned on, and then they're turned off, while other ones are turned back on. And what this is going to do is this will delay fatigue in any given sets of motor units. So some of them are contracting while the other motor units are essentially going to be able to rest for a while. And what this is going to enable you to do is maintain some of these small or weak or sensitive contractions for a very long time, much longer than what would happen if you're using the same motor units the entire time. Now, there's a physiological phenomenon that's going on behind this fatigue, and we'll come back and we'll talk about that a little bit more later on. But this asynchronous recruitment over sustained contractions, just remember, is going to shift the actual individual motor units used when we don't need them all, and this is going to delay how long it takes to fatigue a muscle that's undergoing these sustained contractions. Okay, so that kind of sums up how we are summing twitches together by adding on additional muscle fibers. So let's move on a little bit to the temporal summation of a twitch. So summing up these twitches in time. So even though each individual twitch is all or nothing, each individual these twitches can be summed temporally to give you greater tension out of a single muscle fiber. And this is somewhat analogous to what we were talking about before in the nervous system about summation of graded potential. So in concept, this is going to be similar to that. Now, the reason that this can happen, the reason that this is possible at all, is that the muscle action potentials that are happening are actually a lot quicker than these twitches. And so we can actually get in several action potentials in the duration that a single twitch can take place. And what happens then is if a fiber has not completely relaxed, by the time we get the next action potential, we're going to get some sort of summation that's happening. So when this happens to such a point that the fiber has no time to relax between subsequent stimulations, this is going to cause something called tetanus. Tetanus is when you get these continued stimulations, these continued action potentials, that causes a contraction just to be continual and smooth and give you a sustained amount of force. This is tetanus. So let's kind of look what's going on here and how this is. So down here we have the action potentials, and up here what we're looking at is the contraction strength that is going along with the action potentials that we're looking at down here. So let's look at this first one. We get some sort of action potential along this muscle, and then we get in response some sort of contraction. Now, one thing that I want you to notice is this is not 50 times longer than this action potential. We're condensing down these contractions or these twitches in such a way to make it a little bit easier to see. But one thing I do want you to notice is that this contraction really starts after this action potential has had time to complete. There's this lag, there's this latent period between that. And so what we get is we get this twitch. And if we send another action potential after that twitch is completed, we get another twitch, identical to the first one. However, if we send these two action potentials in such close order that the next one happens before the previous twitch has relaxed, before these muscle fibers have been able to relax from the previous twitch, we will get summation. We will actually get the first contraction, and then we'll get a second contraction that's happening, and it'll actually become a greater force than what we would only have from any individual one. If we do this continually in such a way that they happen very quickly one after another and happen for long enough, what you'll actually see is at some point this will level off. From this one muscle fiber, if we continue to give action potentials, there's a limit to how much force any given muscle fiber can contract with. And once you reach that, and you essentially reach this upper plane that is contracting completely, this one muscle fiber is contracting completely, and it has no time to relax between these contractions, this is tetanus. So, this is this kind of sustained contraction due to repeated action potentials that are so close time, we're not allowing the muscle fiber to relax. And so let's talk a little bit, okay, so what causes this summation? Why do these, in subsequent contractions, why do we actually get an addition of strength? And it turns out there's two major reasons. And the first of these major reasons is that we have this extended period of calcium in the cytosol. And so each action potential going down this transverse tubule is going to stimulate release of calcium from the sarcoplasmic reticulum. And as soon as that happens, as soon as this calcium is dumped down to the cytosol, we're going to start having reuptake of calcium as soon as it's released. So as soon as it's dumped out there, the sarcoplasmic reticulum is hoovering this back up. Well, because that happens so quickly, not all the tropomyosin is going to be moved away for a sufficient amount of time, for a sufficiently long time, in a single action potential for myosin to create a cross bridge with the actin and actually do any work. It's going to not be enough time to create a cross bridge, or as soon as it does, it gets kicked off because the tropomyosin loses, or the troponin loses its calcium, the tropomyosin moves back on and kicks it off, or something like that. However, when we have more action potentials, it means these calcium channels are going to be staying open for a longer amount of time because we are having the voltage-gated calcium channels in the T-tubule continually opening and opening, which means that we have more calcium being dumped into the cytosol. And what that means is we're going to have more calcium in there, so we get longer amounts of time where the tropomyosin is away from the actin binding sites for the myosin, and we get more time that we can actually get this ratcheting effect. The other reason that we're getting summation is that we get stretching of the series elastic component of these cells. And these are just parts of the muscle cell that are stretchy, like a rubber band or something like that. This includes that connective protein that we're talking about, titin, that's connecting these actin filaments, and also other sorts of connective tissue. Well, it takes time to maximally stretch out all this stretchy stuff in these muscle fibers, and you don't get maximal contraction strength until these have been maximally stretched. So think about if you were to hook up some rubber bands to something like a mug or something like that, and you want to drag this mug around your desk with a rubber band. You go ahead and hook up the rubber band somehow to the handle of the mug, and you begin to pull on it. When you first start pulling on the rubber band, there's going to be very little force that is applied to the mug. All you're going to get is stretching of that rubber band. And very little force is going to be applied to the mug. However, as you pull out that rubber band increasingly more, it's going to get closer and closer to its maximal distance that can be stretched, provided you're not breaking it. And as that happens, the force that you're applying to that mug is going to increase and increase and increase till eventually you can drag that mug around by this rubber band. That's what's happening in your muscle cells. As these myosin chains are pulling and contracting by ratcheting up these actin filaments, you got things like the titin that is connecting the actin filaments stretching. And for a while, it's not going to have its full amount of force applied that it could, because this titin is going to be stretching. This connective tissue is going to be stretching. It's going to take a little while for that to reach maximum force. So as we have elongated contractions, or contractions that are happening for a longer period of time, what you're going to see is this force is going to increase as we're essentially taking out the slack out of the series elastic component of these muscle fibers. Now again, that's only going to happen for so long. It's going to happen to the point where you have maximally stretched out these components, and now we're getting full force transferred between the actin and myosin ratcheting and whatever it's applying this force to. So, just in summary of how we're controlling muscle fibers, just to sum this up, we can do this in two different ways. The number one way is we can add the number of motor units that are recruited into contraction. And the second way is that we can increase the frequency of stimulation to these muscle fibers, and that is going to cause summation in these contractions and increase the contraction strength of these muscle fibers until they reach tightness. I talked earlier about how there's these two molecules, both calcium and ATP, that are required for muscle contraction. And I spent quite a bit of time talking about how calcium is being introduced to the muscle cytosol and getting into the sarcomere so that we can get contraction of the muscle. But I haven't talked yet about ATP and how that's happening. So now we're going to talk a little bit about muscle metabolism. And this is really how are we getting the ATP that we need, and how is that being regulated? So ATP is required for muscle contraction. Really, there's these three different points at which this is required. The first one, ATP splitting at the myosin ATPase, is required for the cross-bridging and power stroke that happens to actually ratchet up the myosin along the actin filaments. But we also need the binding of an additional ATP early on to separate the myosin from the actin filaments. So unless we have another ATP to come along and separate that myosin, it's going to stay connected to the actin. Another major place that we need ATP in muscle cells is that calcium ATPase that we need for the reuptake of calcium into the sarcoplasmic reticulum that we need for relaxation of the muscle. So these are kind of three major activities surrounding the basic functioning of these muscle cells that are requiring ATP. So where are they getting the ATP that they need for this? Well, the first major place during contraction that ATP is coming from, so we're getting a donation of a phosphate group to adenosine diphosphate, ADP, from a phosphogen. And these phosphogens in vertebrates, the primary one is this molecule called creatine phosphate. And in other groups of animals, there's a few different ones. So this is the first one of the primary sources of ATP during muscle contraction in skeletal muscle. We also go through oxidative phosphorylation. So citric acid cycle, electron transport chain, have your typical way of generating ATP. This also happens in muscle cells, in the mitochondria of those muscle cells. So this is another major source of ATP. And then one that is just kind of common to all cells, glycolysis. Glycolysis is a way that muscle cells also get ATP. So that's another source. So let's talk about these in turn. So first off, let's talk about the phosphogens. And really, again, like I was saying, during muscle contraction, as whatever ATP stores are being depleted in a muscle cell, the first place that they turn to regenerate that ATP during contraction is donation of a phosphate group from these phosphogens. So the reaction that's primarily happening here is creatine phosphate plus adenosine diphosphate is going to be converted to just creatine and adenosine triphosphate. And this is happening This is a reversible reaction that's happening via the action of an enzyme known as creatine kinase, very standard nomenclature here. But one thing that I want you to notice as we go back here is that this is a reversible reaction. And it's going to follow Le Chatelier's principle in that it's going to be driven by wherever we do not have products in one end or the other. So as we get low on ATP, it's going to tend to drive it this way. And as we tend to get very high on ATP, so if we get lots of ATP, it's going to tend to drive it this way. So during times of high energy, we're going to tend to accumulate creatine phosphate. And during times of low energy, when we're using up our ATP, we're going to tend to take this creatine phosphate and the ADP that we're accumulating and convert it into ATP. So just kind of as a gauge of what's happening here is that resting muscle has about 5 times as much creatine phosphate as it does ATP. So this is actually a fairly robust first source of an additional phosphate group to regenerate our ATP. Another major source of ATP is just your standard oxidative phosphorylation. And I'm not really going to go through any of the details of oxidative phosphorylation. This is something that you covered very in depth back in cell biology. So it's a very deep topic that could take several lectures in and of itself. I'm just going to kind of leave it there. We're making ATP this way. One thing, however, this relies on is it relies on glucose. And so we're relying on glucose that's going to be delivered from the bloodstream, and then also intracellular glycogen. So glycogen is essentially the storage form of glucose when we have it in muscle cells. And so we have this intracellular glycogen that's being stored in skeletal muscle. That's going to be broken down back into glucose and then fed through glycolysis and oxidative phosphorylation to regenerate our ATP supplies as those are running down. Now, there's a few advantages of using oxidative phosphorylation. The really big one is that for every glucose molecule that we shove into this, we're getting more than 30 ATP back. And so this is a very, very efficient mechanism by which we can generate the ATP that we are losing through contraction. Very nice outcomes there. However, there's a couple challenges here that make it difficult for this to be one of our primary ways of getting ATP in all the different kinds of muscles that we have. And the first one is that this requires oxygen. We need to have that oxygen as the final electron acceptor at the very end of the electron transport chain. And so if there is not oxygen available, which sometimes becomes the case during times of very, very high muscle contraction, this can't be met. This requirement for oxidative phosphorylation can't be met, so it's not possible to run it this way. Now, this oxygen is going to be supplied by the blood, so one way that we can increase the available oxygen is to increase capillary supply to a given muscle and inside that muscle, so we have more blood supply to that area. Also, to increase the blood supply, the heart can begin to beat more forcefully, more rapidly. So, as skeletal muscles begin to be more active, you'll tend to get the heart beating more forcefully and rapidly to more efficiently and more rapidly supply large amounts of blood to skeletal muscles to provide the oxygen so they can run oxidative phosphorylation as much as possible. Also, you'll begin to breathe more rapidly and deeply, and we're going to come back to these and talk more about this specifically. I just want to kind of take a time out here and say I'm only going to briefly. There's a lot of physiology that's underlying each of these small bullet points: the supplying of blood, the changing of the beating of the heart, the increasing of breathing. There's a lot of physiology that's underwriting these. I don't want you to think I'm skimming over it, because, well, I am at the moment. However, we're going to come back to these as we start talking about the physiology of the cardiovascular system, as we start talking about the physiology of the respiratory system. We're going to talk about both of these situations, actually specifically in response to high activity by the skeletal muscles, in much more detail when we get back there. So when we get to that point. So skimming over it, just giving a very light treatment right now. We'll come back to this. So the heart can beat more forcefully and rapidly to get more blood to these cells, and also we'll start breathing more deeply and rapidly to better oxygenate that blood as well. Additionally, there's myoglobin in these cells. So myoglobin is similar to hemoglobin that's in the blood, and it holds on to oxygen. And it will tend to help pull oxygen from the blood into the muscle cells for oxidative phosphorylation. So it aids in that, but it can also store some oxygen in the skeletal muscles as well for when it needs that for oxidative phosphorylation. So these are some ways that we can, even though oxidative phosphorylation requires oxygen, and that can be a hard molecule to come by sometimes, there are things that we can do to increase our ability to get oxygen to those muscle cells and increase our ability to undergo oxidative phosphorylation. Because again, we really want to take advantage of this. This 38 ATP per glucose, as long as we can use that efficiency, we want to do that. The other challenge of oxidative phosphorylation is this is relatively slow. It's not proceeding nearly as quickly as glycolysis is or donation of phosphates from phosphogens is. This is going to be a slow way of making the ATP that we need for muscle contraction. So even if we could deliver all the oxygen that we needed, it still might not be sufficient to meet the demands of ATP that we need for rapidly and forcefully contracting skeletal muscles, simply because all of the reactions that take place during oxidative phosphorylation simply take too long, and we cannot get enough ATP made per unit time this way. So those are some challenges, those are some reasons that sometimes skeletal muscles simply cannot use oxidative phosphorylation as a good way to get ATP. So let's go ahead and look at glycolysis next. This is the final way, the major pathway that we could get some more ATP. And one of the big advantages of glycolysis is it's very quick. It has far fewer reactions in it than what we have in oxidative phosphorylation. And actually, we have to run it before we run oxidative phosphorylation anyways. So it's very quick. We don't need to run all these additional of reactions to get our ATP. However, there are some really big challenges to this as well. It's not very efficient. We're only getting 2 ATP per glucose, which means that whatever stores of glucose and glycogen we have in our muscles, we're going to end up running through those stores very quickly and not getting a lot of energy out of them for the energetic molecules that we're dumping in. So this isn't necessarily ideal either. It's quick, but not very efficient. Also, it results in metabolic acidosis, because at the end of this, and the anaerobic endpoint of glycolysis, you end up with lactate. And the problem with lactate is that its pH is very low, and so this is going to perturb a lot of the other biochemical reactions that are happening in the muscles. Now, they have some ways to try to buffer that, but it's still not an ideal situation. So that's kind of the advantages and the challenges of running glycolysis. So let's stop there just for a moment. And all of these pathways of generating ATP, they have their limitations. And so muscles, they're not going to be able to end up running forever. They'll get fatigued at some point. And this can have its origin in 2 different places. The fatigue can be actually centered in the muscle, where we're running out some component that we need in the muscle for contraction, or it can be happening in the central nervous system, where the central nervous system fails to continue to stimulate the muscle as much as it needs to. Now, one thing that you might think would be the case, but is not, is this is not because the muscle runs out of ATP. The muscle does not get fatigued because it runs out of ATP. It gets fatigued before it runs out of ATP. And the reason for that is, or one of the big reasons for that is if you completely deplete your ATP, you couldn't relax your muscles. Because you need that ATP to break the cross-link between the myosin and the actin. And so, if you completely depleted ATP in the muscle, it would go rigid. And so, that's not what's happening. Now, the actual cause of muscular fatigue, that's muscular in origin, is not exactly clear. There's a couple ideas of how this may happen. It could be an increase in ADP signals and causes a feedback loop that causes fatigue in the muscle. It could be an increase in lactate. Remember, this is causing metabolic acidosis, so we could have pH get to the point where it's no longer allowing muscles to contract. We could also be having accumulation of extracellular potassium. So if you remember, as we have these muscle action potentials, we have potassium that is being dumped out of the cell to repolarize it, just like we have in any other action potential. And so we could have this accumulation of extracellular potassium that is inhibiting the ability to continue action potentials in the muscle. It could also be that we're simply depleting our glycogen. Once we run through our phosphogens and we start getting low on glycogen, it could be that this is also another triggering mechanism for a feedback loop that causes muscular fatigue as well. So it's not only muscular in origin, it can also be central nervous system in origin. This is when the muscles, they can still contract. They still have the ability and the resources to contract, but for whatever reason, the central nervous system is not sufficiently activating the afferent neurons to contract. And this is another place where the mechanism is kind of unclear. We're not exactly sure. So, muscles do become fatigued, and this can be of two different places, muscular or central nervous system in origin. While we are undergoing these very large and very long major bouts of using and highly contracting skeletal muscles, there will be afterwards, even after you are done with that contracting, you'll probably have noticed that there'll be some heavy breathing after that. You'll be continuing to breathe rather heavily after strenuous activity. Why in the world is that? Why do your muscles continue to demand oxygen even after contractions have stopped? That's because we've had an oxygen debt, what's often referred to as an oxygen debt, that's accumulated by muscles. And what happens here is that when you get oxygen derived from non-oxidative sources, so when we're using the donation of a phosphate from a phosphogen, or we're running glycolysis with an anaerobic endpoint, what happens is that we need to reset ourselves using oxygen. And this is the reason why. We need to repay that. So one of the first things is that we need to replenish the ATP by oxidative phosphorylation, and much of this is going to be used to replenish the phosphogens that we ran through early on in muscle contraction. So we need to replenish those phosphogens. We need to do that by making ATP that we can then, through creatine kinase, rephosphorylate creatine. And this is going to happen by oxidative phosphorylation. Therefore, we need to be using oxygen to run oxidative phosphorylation. Also, the lactate that's been built up needs to be converted back to pyruvate so we can then run this through the Krebs cycle so that we can, undergo oxidative phosphorylation again. So this also is going to require oxygen. So even after we have finished with contractions and finished with a lot of these highly anaerobic metabolic processes, we still need to repay this oxygen debt and essentially reset ourselves to the state that we were in before we underwent this strenuous activity. And a lot of that's going to require oxygen. So we need to repay the oxygen debt that accumulated during anaerobic sources of gaining energy. All of this being said, skeletal muscle contraction is really energetically inefficient. This isn't true of all muscles, but specifically very true of skeletal muscle. It actually turns out only 25% of the ATP energy is actually converted into mechanical energy of the contraction. So very, very little of the overall energy from that ATP goes directly into that. Now, The rest of the ATP energy, the majority of it, 3/4 of that ATP energy, 75%, is converted into heat. Generally, this isn't great. This isn't what you want to be happening. Now, sometimes this is a good thing. If you'll notice, sometimes if you're outside, scantily clad on a cold day, or not clad enough, you might shiver. And that's taking advantage of this situation that skeletal muscle contraction actually makes a lot of heat. And so you can, at times, purposely generate a lot of heat by skeletal muscle contraction. However, as you probably may have heard about at certain times, this can be a bad thing as well. So, for instance, a couple summers ago, there was quite a bit in the news of several high school students collapsing because of hyperthermia during football practices in late summer. Now, this is a direct result of this high contraction or high rates of contraction of skeletal muscles. That's creating a lot of heat, and if it happens to be in a thermally rich environment already, you don't need that heat. A lot of this heat can begin to accumulate in the body, nowhere for it to really go, because there's not much of a temperature gradient with the outside, and this can become a real problem. So sometimes a lot of ATP energy being converted to heat is a good thing when it comes to skeletal muscles. In fact, often it is, because it's one of the major ways that we're actually heating ourselves. Not all the time, though. So, now that we've talked a little bit about how the metabolism of skeletal muscle tends to work, and the different ways in which skeletal muscle can gain its ATP and gain the energy that it needs to drive these contractions, we can start to begin to talk about specific types of skeletal muscle fibers that are employing different, or optimizing, I should say, different strategies for obtaining ATP. And the different ways, the different types that we're going to be talking about are, number one, these slow oxidative fibers, which are also known as type 1 fibers. Fast oxidative fibers, which are type 2A fibers. And then also fast glycolytic fibers, which are known as type 1X. Sometimes also referred to type 2B. Excuse me, I might have said type 1X. I meant type 2X. There's type 1, the slow oxidative, type 2A, the fast oxidative fibers, and then type 2X, or sometimes referred to as type 2B, the fast glycolytic fibers. So let's talk about what differentiates these different fibers. First, I'm going to talk about slow versus fast. Then next, I'm going to talk about oxidative versus glycolytic fibers. So, fast versus slow. And really, this is pretty intuitive. One is fast, one is slow. Fast fibers have a higher myosin ATPase activity. So the myosin ATPase on the myosin head is a much more active version of the myosin ATPase than is in the slow version. And contraction time, now remember, contraction time is the time from beginning of a contraction to the peak peak tension of a twitch is only 20 to 40 milliseconds. Now, if you remember when I was talking about contraction time earlier, I was giving you the average of 50 milliseconds, and for all muscle fibers, that is approximately right. However, for the fast fibers, we're actually talking somewhere that centers on about 30 milliseconds. So a bit faster than the average I was telling you before. From onset of contraction to peak tension of contraction, 20 to 40 milliseconds. So, pretty fast. Slow fibers, however, and we're just talking slow in relation, still a very fast action, have this lower myosin ATPase activity, so that myosin ATPase is not nearly as enzymatically active. And contraction time here is 60 to 100 milliseconds. So considerably slower, somewhere centering on or averaging about 75 milliseconds then. Excuse me. Yeah. Somewhere in that neighborhood. So, yeah, so that's these are considerably slower fibers, and that's really is really intuitive. That's really what's just going on here. Let's now talk about oxidative versus glycolytic fibers. Now, oxidative, again, this might be pretty intuitive. Oxidative fibers tend to rely more heavily on oxidative phosphorylation, even despite the challenges that oxidative phosphorylation has. So these are going to be fibers that are specialized for certain kinds of activities. And one of the really good advantages of these oxidative fibers is that they're not going to readily deplete their energy stores. They're actually going to take quite a while to use them up, because the phosphogens are abundant in both types, but then the glucose that we have and the glycogen that we're converting into the glucose, we're not going to run through nearly as quickly, because for each one of those, we're getting over 30 ATP out, rather than just simply 2 ATP we're getting by only running glycolysis with an anaerobic endpoint. They also have a higher mitochondria volume. So, as you will probably guess, because mitochondria is where Krebs cycle and electron transport chain take place, we have a higher mitochondria volume in these oxidative oxidative muscle fibers. We also have a higher myoglobin volume. And this is going to result in a red color, just like blood is red due to the hemoglobin in it. Oxidative fibers tend to be very red because of their high myoglobin content. And this is going to do a couple things. So myoglobin is going to be pulling oxygen out of the blood, so helping fuel oxidative phosphorylation by supplying it with a good amount of oxygen. But they can also hold on and store some of that oxygen, so that's another way that we could be helping out this process along as well. So oxidative fibers are going to be fibers that tend to be adapted for high endurance running contractions for a very long time. That's what we tend to use them for. Okay, so let's talk then about the glycolytic fibers. And again, fairly intuitively, glycolytic fibers tend to rely more on glycolysis. And these fibers will tend to contain a lot of glycogen. Again, we're going to be running through these energy stores very quickly, because we're not getting much energy out of each one, out of each molecule of glucose. So they need to have more of it in order to run contractions for very long at all. So these fibers are going to tend to have a lot of glycogen. They're also going to have fewer mitochondria and myoglobin, so unlike the oxidative fibers, which tend to appear red, these glycolytic fibers are going to tend to look white. And generally they're larger in diameter. They're larger in diameter because they contain more actin and myosin filaments. So they're containing more thick and thin filaments. And this is going to generate higher tension. These can actually contract more strongly, generally, than oxidative fibers can, because of this increased amount of actin and myosin. So again, they can generate these powerful, explosive contractions. They can be using up this energy very fast. You can kind of think about this as a muscle car or something like that is very, very gas inefficient, but can go very fast with a lot of power, just not for very long. And like I said, they're going to exhaust their fuel very quickly because they're not very efficiently using the fuel that they have. They're not getting a lot of ATP out of that. So let's take a look. Actually, fish muscle fibers is really easy to see this. This is tuna here. And tuna are large fish that swim at very high speeds for a very long time in the open ocean. And because of that, they tend to need to have very high endurance muscles, because they're going to be working at high levels for extended periods of time. And so they tend to have a lot of very dark red meat. And so you'll notice up in here, this all is red through here. That's because of the very high amounts of myoglobin that is in that tuna muscle. On the other hand, this white chunk of meat up here, this is halibut. And halibut has a very different kind of lifestyle. So halibut tend to be living on the seafloor, and they're sit-and-wait predators. So they are spending a very long amounts of time sitting just on the bottom, waiting for prey to come by. When the prey finally does come by, halibut and other flatfish will catch it in a very explosive, quick manner. But after that, then they go back to resting again. And so they have muscles that do not need to work for very long periods of time, but when they do, it needs to be very quick and explosive contractions. Underneath here, what you're getting right here, this is salmon. And salmon is more like tuna. They're pelagic fish that tend to swim very often. Now, they're not swimming nearly as quickly as the tuna are, so you'll notice it's not nearly as red meat, but it is quite a bit redder than what the halibut is. And these muscles are much more adapted for high endurance performance than the halibut is. And what you'll see down here is a small piece of broccoli or something else like there. It's green because it has chlorophyll in it, and it's not an animal, so we're not talking about the physiology of this plant whatsoever, because compared to animals, plants are relatively unimportant and no one really cares about them. What you're seeing here is some muscle of Nile perch, and one interesting thing here is that you actually get to see both red and white muscles in the same set of muscles in an organism here. Fish tend to segregate their red and white fibers, so it's actually fairly easy to see where the red fibers are. So kind of in here towards the middle of these fillets, and on the outside we tend to get these white fibers. And so oftentimes or fish specifically will segregate white and red muscle. So again, like I just said, the arrangement of these fibers in some types of vertebrates, like fish, are going to be segregated. You're going to have your glycolytic fibers in one area. You're going to tend to have your oxidative, fibers in another area. However, that being said, in terrestrial vertebrates, most terrestrial vertebrates, these are mixed in. So all of these different fiber types are going to be mixed in together in the same muscles. Now, various muscles can vary densities of the relative numbers of these different kinds of fibers that they have, but they'll still be mixed in. So one thing to think about here is like white and dark meat in poultry. So the types of birds that poultry are, galliform birds, they tend to live on the forest floor and only use flight as an escape mechanism or to get them up into trees to roost or something else like that. They're not flying for very long distances. So the flight muscles in the breast are very white because they just need, short bursts of activity. However, the leg muscles that hold up the animal for long periods of time, they use for walking, are much darker, or when you see them alive, redder, because they have a much higher density of these glycolytic fibers. Now, neither of those two muscle types are purely one type or the other. It's a mixture of the two. And they're not tending to segregate them like you saw in the Nile perch picture just a moment ago. They're all mixed together. Now, one thing I should add in here is that each motor unit, so each motor neuron plus its associated muscle fibers, are going to be one type of fiber. They're either going to be glycolytic, or they're going to be oxidative. And in fact, they're going to be either slow or they're going to be fast. They will not be a mixture of both slow and fast in one single motor unit. So, that being said, muscle cells can adapt. Even though a lot of muscle cells are specialized, one or the other, there's a little bit of a continuum between the glycolytic and the oxidative. But there is no continuum between the slow and fast. You're either slow, you're fast. There is an ability of muscle cells to adapt from one type of fiber, for instance, an oxidative fiber, to a glycolytic fiber, vice versa. And one way this can happen is that you can increase your ATP synthesizing capacity. So a glycolytic fiber can become more oxidative, and this can happen a couple different ways. It can increase the number of mitochondria that are inside that muscle fiber, and then also there can be an increased blood supply by adding additional capillaries to supply given muscle fibers. And so that way we can get an interchange between oxidative and glycolytic fibers. Additionally, we can have hypertrophy, where we're taking fast glycolytic fibers, and they can add myosin and actin filaments, and this will actually cause an increase in the diameter of these glycolytic fibers. And this is happening over a period of time, and it'll add on to what's additionally there. However, what cannot happen is there's no transition that can happen between slow and fast fibers. Now, you can change their relative abundances at times, but a single muscle fiber is either going to be slow or it's going to be fast. And one big reason for that is that the neuron supplying it is going to be specialized for the kind of muscle cell that it is. The neuron is either going to be a slow muscle neuron or it's going to be a fast muscle neuron that'll act quicker. And so, in order to be able to change those, your body would have to be changing both the nerve cell and the muscle cell in concurrence, and that doesn't really happen. So, you don't really get interchanges between slow and fast type of fibers. So really, that's going to finish up with the metabolism. We have just a little bit more that we're going to talk about with muscles. We've been, up until this point, essentially just talking about skeletal muscle, but from here on out we're going to be moving into a different muscle type, specifically smooth muscle, and that should really finish up what we're talking about with muscle.